So in the next question, we're given an alignment with the secondary structure annotation, a reference structure, and we're being asked to draw each of the secondary structures of each of these RNAs. The reference structure isn't really needed, but it's useful to look at all the same. The secondary structure is encoded in the information here. Remember that for each left-facing parenthesis, there's a right-facing parenthesis, and vice versa. Whatever is in this column pairs with whatever is in that column, this column with that column, this column with that column, this column with that column. And all you have to do is draw each one sequentially. So, let's start with the first one. The first base pair is this G with this C. The next base pair, this G with that U. Remember that UG pairs are perfectly good in RNA structure. Next is this G with this U. And last is this G with this C. And the loop is G, A, U, A. And the tail is UA. And you can see that these two structures are very much alike. I'll erase this just to make some space. The next one, you can also draw it this way. You have 4 in a row. We'll just go A, U, G, C, and those are paired with UA, C, G, reading backwards. U, A, C, G. The loop is U, U, C, G, and there's a UA tail. And we're done. The next one reads G3Us, U, 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 it's this. The complementary strand reading forward is A, G, C, and the loop is just 3 U's. The gap here means that this loop is one shorter than the other two that we've seen so far. And there's, again, a UA tail. And that one's finished. This one's a little different because you can see that there's only 3 base pairs here instead of 4. So we'll go this G with this C, this G with this C with this G. There's a gap here, and of course a corresponding gap here. This is missing a base pair, and so both partners of the base pair have to be missing. This means this is a good alignment. The loop is C, U, U, G, and the tail is a single A residue. That U in the other sequences is absent from the tail in this particular example. And we're done with that one. GGC paired with GCC, CUUG loop, A tail. This last one is a trivial example just to show you that there can be no sequence similarity between any of these, and you can still align them based entirely on the structure. This is just a string of U's and a string of A's, but it's laid into the same structure. You have UA, 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 UA. You have U, 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 U as the loop, and A as the tail, and you're done. And so you've aligned these things based on the structure, not on the sequence. There's no sequence similarity between this one and any of the others, and there's not a lot of sequence similarity between any of these. And yet, because the structures are the same, you can align them, and because you have the structural alignment, you can draw.